Hello, this is a lecture for Affinity Chromatography. We've recently talked about ion exchange and gel filtration chromatographies, and this is the third installment with Affinity Chromatography. In Affinity Chromatography, there are two partners, the protein of interest that you want to purify, and a molecule that interacts with that protein that is covalently attached to a bead of a specific matrix. The diagram on the top left is a schematic of affinity chromatography. We have a mixture of proteins that are red and yellow with different shapes and sizes and binding properties. The red is the protein of interest that is being purified with this particular affinity chromatography. The bead is in gray and is made up of a specific matrix, and the black is the specific molecule that interacts with the red protein that is used to capture the protein of interest. All of the other molecules do not interact with the covalently attached molecule and thus flow through the resin, leaving the red protein behind attached to the beads. There are many different partner pairs between biomolecules and molecules that they bind or can be used for affinity chromatography, and several are listed here at the bottom right. So you can have enzymes and you can have their inhibitors or cofactors covalently bound to the matrix. And you can have proteins that bind specific hormones and vitamins, and those can be attached to the matrix. One that we'll be using in lab is a particular chelator of a metal ion that interacts with a peptide that has been genetically engineered to be N-terminal of your protein of interest. In the next two slides, I'd like to outline some common terms used in affinity chromatography. First is the matrix, which I've mentioned before. This is what the resin is made of and what the ligand is chemically attached to. The ligand usually is not directly attached to the matrix. There's typically a spacer arm, which is a chemical moiety that brings the molecule that is covalently attached slightly away from the matrix surface. We often call the molecule that's being bound to the resin that will then bind to the protein a ligand. Here that's shown as a blue triangle on the top of the chemical spacer that's attached to the matrix. Here are a few more terms used in affinity chromatography. In fact, the first three terms are used for any chromatography. When we talk about binding, we're talking about a condition in which the protein that is going to be purified can interact with the ligand. And so when we load the protein on the column, we're binding the protein to the ligand. For most affinity chromatography types, the buffer conditions are very important to facilitate that binding event. Elution is also a common term in most chromatographies, and that is the buffer conditions in which you can reverse the interaction between the protein you're trying to purify and the ligand that's on the matrix in order to remove the protein after capturing and removing the contaminant proteins. The elution happens after the wash, which is the next term. The wash is the step in which, after you bind all of your molecules in the mixture to the column, you can then wash the proteins that don't interact with the ligand on the column off the column, and so that all that's left behind is your protein of interest. The last two are specific to affinity chromatography. Ligand coupling is if you want to create your own affinity chromatography. There are different ways that you can chemically couple the ligand to a pre-activated matrix, which is the next term. So they are matrices that have been chemically modified to facilitate a coupling of a specific functional group on a ligand, and we'll talk more about these specifically later. There are 4 steps to most chromatographies. The first is equilibration. That is, you equilibrate your matrix with a buffer that is compatible with your protein of interest and the binding event. Ideally, your protein should already be in this buffer. Next, you load your protein onto the matrix and then wash with the appropriate buffer that would wash contaminant proteins off the resin but keep your protein of interest on the resin. Third, you elute. Elution is done in many different ways. It can be done by changing pH, changing salt or ionic strength, or by adding a molecule that will compete for the binding site on either the protein or the resin. Once the protein is eluted, most resins can be reused and should be re-equilibrated for use again. Here's a general chromatogram as would be recorded on an FPLC with the UV 280 absorbance versus column volumes. First, you can see there's an equilibration step of 1 to 2 column volumes of buffer. Then there's the addition of your sample that is a mixture of proteins, and there's an increase in A280 due to all of these proteins being loaded onto the column. Then you have a wash step where you see the UV coming down, where protein contaminants that aren't interacting with the matrix are washed off the column. And then you have the elution step, which is several column volumes, and you can see when you change to the elution buffer in this chromatogram a sharp peak in which your protein is eluted. This slide shows 4 types of elution methods. Method 1 is a simple change in buffer composition which allows elution to occur or disrupts the interaction between the protein and the ligand on the matrix. For example, for a GST fusion, you may just change the redox potential in order to elute the GST from the glutathione ligand. The second method, probably the least preferred method, is to go to extreme pH or denaturant in order to disrupt the interaction between the ligand and the protein. Method 3 is showing a competition elution in which something is added that interacts with the ligand on the matrix to compete for the interaction of the protein with the ligand. The complementary but opposite elution is one in which you would add a molecule that binds to the protein and competes for the binding site on the protein rather than the ligand. Just as in ion exchange chromatography, you can set up your elutions of affinity chromatography in a stepwise fashion, such as shown on the left, or a gradient fashion shown on the right. You will be doing a step elution in the laboratory and would need a gradient mixer or an FPLC to generate a gradient elution. Be sure you understand these chromatograms and how the step and gradient elutions are represented. One easy way to utilize affinity chromatography for any protein of interest is to use fusion tags. That's when a protein that has a known binding affinity is genetically engineered either N or C terminally to the protein of interest. This is usually done in the plasmid, and many commercial expression vectors have fusion tags already in them. Common fusion tags are his tag, such as the one used for your proteins of interest, streptavidin, and GST. You'll be using metal interaction affinity chromatography, or IMAC, in the laboratory. On the left are 2 types of metal interaction affinity chromatography resins, nickel NTA and nickel IDA. The divalent metal is chelated by the resin. In nickel NTA, you can see the atoms that are chelating the nickel, 4 of them, 3 oxygens and a nitrogen from the matrix. In nickel IDA, there are 3 atoms chelating the nickel, leaving 3 chelated waters. When the his tag is bound to the resin, the imidazoles are chelating the divalent metal, such as that shown on the right. In the laboratory, you will follow these steps for affinity chromatography. You will lyse your cells through freeze-thaw cycles and enzymatically with T4 lysozyme. You will isolate the cell debris using a low-speed spin, which is not shown on this flowchart, and you'll take your supernatant and load it on your nickel NTA resin. Your protein should bind to the resin, and other proteins from E. coli, shown here as the light blue proteins, will wash away. Your protein that has the his tag, which is represented here in red, will bind to the resin and should stay on the column during the wash. After the wash, you will elute your protein with a concentration of imidazole around 200 millimolar. During the elution, you are displacing the histidine residues that are chelating the nickel with imidazole. Shown on the right are the structures of imidazole and histidine, showing that the chemical moieties important for chelating the nickel are identical. Therefore, imidazole is an efficient elution molecule. On the bottom is shown a gel and a chromatogram of a typical IMAC purification. On the gel, you will see the molecular weight marker, the cell lysate that's loaded on the column, the flow through, which is the volume that is collected while loading the column, a few fractions of the wash where you can see contaminants as well as the protein of interest, and 2 elution fractions from the elution profile, which has the protein of interest. The gel also allows you to assess the relative purity of the elution. That is, how many other contaminants can you see and how intense are they compared to the protein of interest band? There are other ways to use affinity chromatography beyond fusion tags that utilize the protein of interest's natural ligands or inhibitors, or utilizes antibodies that have been raised to that particular protein. This approach requires one to use chemistry to attach ligands or inhibitors or the antibody chemically to the matrix. There are many matrices that have activated functional groups that can be used to attach molecules of interest to them. For instance, to purify a serine protease, which you've studied in biochem lecture, you could couple the benzamidine inhibitor to a spacer arm attached to the Sephrose matrix. This could be used to capture the serine protease on the resin. Alternatively, if you were to be purifying trypsin, which recognizes arginine and lysines, you could couple arginine to the matrix through a spacer arm. Here's another example using a cofactor. Many dehydrogenases use NADP as a cofactor in their catalytic reactions. If the cofactor is not absolutely important for the structure of the protein, then you can use the cofactor bound to the resin to capture the dehydrogenase protein of interest. There are a lot of different activated resins that you can commercially buy. Some of them are shown here in these tables. They differ in their length of spacer arm and the reactive groups available. There are 4 sections to this table. There are groups that are found on proteins such as amino, carboxyl, and thiols. There are groups that are available on sugars such as hydroxyl, amino, and carboxyls, polynucleotides, and coenzymes, cofactors, antibiotics, and steroids, basically ligands. So if you had a protein, you may want to attach your protein via the thiol and the cysteine groups, and you could use any one of these 3 activated matrices depending on the length that you wanted to have from the matrix surface, 4 atoms, 10 atoms, 12 atoms, and depending on the chemistry that you wanted to utilize and its efficiency. This is just to show you the variety of chemistries available in order to create your own resins. This slide is just showing you one of the specific coupling reactions available for amino groups utilizing an N-hydroxysuccinimide activated Sephrose resin. Finally, I want to end with the general structure of one of the most common matrices used in affinity chromatography, Sephrose. Sephrose is a brand name that is derived from separation pharmacia agarose. Sephrose is a cross-linked bead form of a polysaccharide polymer that is predominantly made up of agarose. The partial structure of agarose is shown on the left. A schematic of the cross-linked polymer is shown on the right. This mini lecture has provided you the basics to affinity chromatography. It has described the practice of using fusion tags to direct an affinity and the process of utilizing the properties of a particular protein and designing a resin that is specifically targeted to a protein property, such as a substrate or cofactor.